Welcome back, everyone. So we continue talking single cell data analysis, and we will continue with integration and batch correction for single cell data analysis. So first of all, there is a little bit of a Wild Western terminology, and I suggest to use the terminology introduced by Ricard Argilago and the colleagues in their, I believe, Nature Biotechnology paper from 2021, where they introduced three different types of integration: horizontal, vertical, and diagonal. And I'm showing horizontal and vertical integration, and I'll come to the diagonal integration in a few slides. So first of all, horizontal integration assumes, so both types assume that one dimension in these matrices that we are combining, one dimension is constant across different data sets. So for example, for horizontal integration, the features are the anchors, what they call anchors, meaning that gene names are the same across all different data sets. So you essentially, you're working within a single omic. You're taking maybe RNA-seq, single cell RNA-seq data set, gene expression data, and you're just combining different data sets across different labs, across different, maybe technologies, countries, etc. But it's the same RNA-seq. So it's basically the gene names are the same across all these three different data sets. This is what I believe is very similar to what was called previously batch effect correction. When we do this horizontal integration, this is what was called in the old days batch effect correction. Now it's called integration. And I believe often, in most of the case, I would say, when single cell people do integration or say they're doing integration, they assume this type of integration, horizontal integration. So integration within single omic across different batches. So this is very different from vertical integration, where the cells are the anchors. So here the features can be, the biological variables can be completely different. It can be, CpG sites and genes and, ataxic peaks, chromatin accessibility information. But measured, so all these three types of information is measured in the same biological cells. So the cells should be the same, just the variables, biological variables measured in them can be different. So that's what is called vertical integration, and it's a little bit more challenging problem, because I'm not saying that batch effect correction is a simple task to do, but at least conceptually you understand what you're doing. You understand what kind of sort of outcome you want to get. While in vertical integration, sometimes it's even difficult to wrap your head around this. So what do I mean by combining CpG sites and gene expression and chromatin accessibility? So what does it mean? So what is the output? Why am I doing this? What do I want to get? So I'll cover both of them, and I'll try to explain and share what I know. So we start with horizontal integration, or what I call it, integration across cells. And so here the problem is the following. Imagine that you have cells, you have a mixture of cells coming from three cell types and two batches. So maybe you are visualizing these six cells through a PCA, through dimensionality reduction. And here I'm coloring cells by their type. So there are green cells, maybe there are these orange cells and blue cells, three types of cells, but we see that they're clustering with respect to something else. So these two clusters visually, what we can detect visually, they're not based on cell type. but they probably have some technical origin. They come from maybe two different batches. But we are talking about gene expression here. So the point of integration is actually to come to this PCA plot where cells are grouped by their cell type rather than technical factors. And so to do it, in the simplest way, what Lim or Combat did previously in the old days. So we are creating the following model. We are modeling this gene expression matrix. So these 6 cells are coded here in this gene expression matrix with 5 genes. And we are creating the following model. We assume that the variation in gene expression in this matrix is due to, first of all, batch. Second, due to different cell types. So we are constructing these 2 variables, green and orange. And here I'm sort of coding my cells C1 and C2 and C3, for example, belonging to the batch number 1, this one, so C1, C2, and C3, they will belong into the batch number 1. And the rest of the cells, they will belong into the batch number 2. This is what 1 and 2 in this vector mean. And the cell type, so here I'm saying that cells number 1 and 4, they belong to the same cell type. And cells number maybe 2, and 5, they belong to the second cell type, etc., etc. So this is what this vector coding in this vector means. And our goal, the goal of this modeling is to get rid of the batch variable. So we want to do some sort of transformation with our data that brings this coefficient, the prefactor beta, brings it to 0. And we want to actually keep the second variable. Because this is our variable of interest. So here is a caveat. So we want to. So this will work. This will work only if the batch variable and the cell type variable are uncorrelated or weakly correlated. Otherwise, if imagine that these batch and cell type variables are correlated, so bringing one variable to 0, you'll automatically get rid of their second variable as well. So ideally, in an ideal world, we believe so. One of the major assumptions of batch effect correction procedure is that your batch is uncorrelated, or what they say orthogonal, to your variable of interest. That's a major assumption. You never know this, but you assume. You believe that this is the case, and in a good experiment it should be the case, but sometimes if it's not the case, then you can run into problems. And here, this is what people typically do, or what they did working with bulk data before the single cell era began. So this is how people corrected their batch for bulk RNA-seq, for example. And here you can see immediately a difficulty. So yeah, I explicitly coded all the cells to belong to a certain cell type. So how do I know that my cells number 1 and 4, they belong to the cell type number 1? Well, it turns out that this information is actually available in bulk sequencing, where you, for example, analyze sick individuals and healthy individuals. Then this variable of interest is your phenotype of interest. It's actually almost always known. This is how you do differential gene expression analysis. You're comparing 2 groups, and the coding of the group coding is actually known. This is your variable of interest. You want to keep this phenotype of interest and get rid of the batch. While in single cell, we actually don't have this variable. So that's a complication. So this variable is actually what we want to learn when we are doing our single cell gene expression analysis. So how to ensure that removing this batch variable actually will not lead to the removal of valuable cell type information. And this is why basically Combat and Lima and this simple regressing out the batch strategy doesn't work for single cell really, or has limited applications. So you still can hope for the best, and try to remove the batch variable just to regress out the batch. The batches variable is usually known. You know how you generated your data. But then, of course, there is no guarantee that it doesn't affect the valuable cell type information. So that's why this regressing out should be taken with caution. And there is a kind of a proxy, a replacement for this cell type variable, which is specific to single cell data, which is a cluster assignment. So basically, you you create another variable that assigns all cells to certain clusters, and you proceed with batch variable removal, approximating your cell type variable with your cluster assignment. So you believe that your cells are assigned to a certain cluster according to their cell type. And not according to batch. That's a major assumption, but still it's what in principle you can try to use, and this is what Harmony uses. So basically it utilizes this kind of proxy, saying that okay, I approximate my cell type variable through cluster assignment, and I'm trying to to decorrelate, to make those two batch variable and cluster assignment uncorrelated. So as little connection between those two guys as possible. Yeah, so that's a strategy and explanation why this batch effect correction. I said it's kind of an old problem. You conceptually understand what you're doing. You want to get rid of this variable and keep the biological variable. So the problem is well formulated. However, this complication in single cell that you don't know what cells belong to what type, and this is exactly what you want to discover in your analysis. This actually makes batch effect correction problem less trivial, and here you need to think a little bit and put more intelligence into your analysis. And this is what was one of the major goals in Human Cell Atlas consortium many years ago. They attempted to create a comprehensive atlas of human cells from different organs and tissues. Many big names in the field were recruited into this consortium, and they declared that the data harmonization and integration was one of the major challenges of the Human Cell Atlas. And so some analysts like Raul Satij and John Marioni and etc. These guys started thinking how to solve this data harmonization problem and came up with a few methods. So one of the first methods in the field, which was kind of single cell RNA-seq specific, was what is called mutual nearest neighbors, MNN, from John Marioni's lab. And the idea is interesting. So if if you have clusters of cells in two different batches, the way you correct for the batch effect, you check, you find a pair of cells that are the nearest neighbors across the batches. So you find two closest cells across the batches, and then you say that I believe those cells, the nearest neighbors across batches, they belong to the same cell type. And that's a huge assumption. But this is how it works. So you believe that those two guys, nearest neighbors across batches, they should belong to the same cell type. And making this assumption, you are saying, okay, they should be placed in the same cluster, but they're not. So this separation is artificial. Let's remove this separation. Let's renormalize, all the distances here. Let's renormalize gene expression in such a way that there is no separation between those two nearest neighbors anymore. And this will lead to renormalization and correction for the distances for all other cells. So you create this kind of correction vectors, and you renormalize, recompute gene expression for all the cells, and this is how you kind of end up with a harmonized data set, with a single harmonized data set. And here they're listing very important assumptions of this method. And as I said previously, so one of the biggest assumption is that the batch effect is almost orthogonal to the biological subspace. And the batch effect variation is much smaller than the biological effect variation between different cell types. Because, as you can imagine, these two nearest neighbors across batches, they can be, nearest neighbors as a consequence of strong batch. So it's not necessarily biological reasons why they look like nearest neighbors. They can be, artificially placed in these positions to be nearest neighbors. It's a manifestation of the very, very strong batch effect. But if batch effect is weak, if there is this decorrelation or absence of correlation between biological subspace and technical batch variable, then this probably will work. And in practice, it actually does work pretty well. So this was kind of the method number one in this field, single cell specific method, going beyond, combat and lemma and all these bulk RNA-seq correction methods. So then Surratt suggested what is it called? Cross anchors. No, it was called canonical correlation analysis method. And I kind of add this canonical correlation analysis plus dynamic time warping, because the dynamic time warping was an essential step there. So essentially, what Surratt suggested to do, they said, okay, if you have two matrices, you can always apply what is known as PLS, partial least square regression, or like canonical correlation analysis is kind of almost a synonym for partial least square regression. So it's the same thing. And the idea is that you construct a covariance matrix. So if you have two matrices X and Y, you can construct a covariance matrix, another matrix, which is kind of, you can think about it as a correlation between X and Y. And you can use this covariance matrix for, matrix factorization, similar to what PCA does. PCA actually, it doesn't have two matrices. It has just one matrix X. And it says, okay, let's compute a variance. So you can't compute covariance, but you can compute a variance of matrix X and proceed with the problem, linear algebra problem, which is called eigenvalue decomposition. So basically, to build these eigenvalues and eigenvectors, that kind of, in this particular case, when you deal with covariance matrix, so essentially, intuitively, you can think about these eigenvectors computed in this way as a common axis of variation. So you project your data sets to some axis. You construct latent variables, what you call common axis. That kind of they contain common information, common variation across X and Y. And this is what canonical correlation analysis does. However, they realized that maybe this canonical correlation analysis was not, perfect. Looking at this picture in the middle, you can see that these blue and or green and red cells, they're kind of, they're not perfectly merged, and that's why. But they've kind of showed similar pattern. They both are kind of triangular looking, and in addition, Surratt suggested to apply this dynamic time warping procedure, which I basically familiar with dynamic time warping in context of time series data analysis. So in time series data analysis, dynamic time warping is used for as a distance metric between trajectories. So you don't have data points in time series data. You have trajectories, and then how to compute a distance between, two wave functions. So here I'm showing two wave functions, the orange and the blue, and I compare them with this straight line, the green line. And if you compute, Euclidean distance was shown not to be, perfect. Or it didn't make sense that if you compute Euclidean distance, it turns out that a wave function, for example, this blue wave function, is closer to the straight green line compared to the distance between two wave functions. So it's kind of counterintuitive. You would think that two wave functions should resemble each other more than a wave function and a straight line, but not in terms of Euclidean distance. Indeed, Euclidean distance was kind of shown to be not optimal for time series data, and that's why people came up with this dynamic time warping definition, which is kind of it's algorithmically it's difficult. I think it's a recursive like series of very, very complex equations. But the idea is that after you sort of compute this dynamic time warping distance, everything suddenly makes sense. So indeed, two wave functions, they have smaller distance with respect to each other than the distance between them and the straight green line. And this kind of sometimes interpreted by people as if you squeeze two wave function and make them similar to each other as much as possible through stretching or compressing two wave function, and you get this effect of a high similarity between two wave functions. So anyway, this was a dynamic time warping, and Surratt suggested to use this. At first glance, it's completely unrelated. How is it related? This wave function, how is it related to data integration? So it turns out that this kind of stretching, I said that dynamic time warping algorithm is usually interpreted as stretching and compressing of different time series data. And they basically suggested to, stretch a little bit this if you look back at this CCA, canonical correlation analysis plot. So if you a little bit, stretch and compress these two clouds of points, the red cloud and the green cloud, they will be overlapping almost perfectly. They will resemble each other much better. This is what they kind of used, dynamic time warping. They used on the top of CCA and came up with quite nice harmonization of data within a single omic across different batches. Then a few years later, there were two major releases. Two methods came, more advanced methods, and slightly different from CCA, because basically CCA and MNN, I would say those methods are kind of, you can find them in data science and in different applications. It was not really single cell specific methods, and they use kind of known ideas. Harmony was a little bit different. Harmony has a funny algorithm that tries to mix all different clusters across batches as much as possible. So basically it implements some, smooth, what they call smooth assignment of cells to clusters, or basically like moving cells between clusters and running k-means, basically clustering all the time, and having a special penalty function that prioritizes mixing of different batches within each cluster. So what you want to achieve, you want to have all batches to be represented in all clusters. So this kind of mixture is implemented through this. So if you check the supplementary in the equations, they specifically say that we introduce this additional term in our cost function, because first two terms in this cost function, this is actually k-means. So indeed k-means can be reformulated through this optimization problem But then they kind of added this additional term, additional penalty to the cost function, which is supposed to harmonize or kind of to mix the function below penalizes clusters with low batch diversity for all defined batch variables. So basically, clusters with low batch diversity are not preferable from the point of view of this algorithm. So that's actually kind of the whole idea. It's purely algorithmic. It does not have much to do with equations, and it's a little bit tricky, a little bit black box to me. From all these methods, I think I understand harmony the least because it's like a little bit strange why you want to mix. It's not always makes sense to me that you want to mix all the batches in all clusters. So another major method in the field was Scanorama, which I understand basically as kind of MNN. So it's like a mutual nearest neighbor algorithm, but generalization for multiple data sets. So the original MNN, it actually worked with two data sets, the query and the reference. And it didn't work. Actually, it was not straightforward to generalize it to multiple data sets. And this is what Scanorama achieved. And they kind of have some improvements of MNN. For example, they say we do not require at least one cell type shared across data sets. And this was actually one of the, if you check, I don't know, here, one of the assumptions. Somewhere here they were writing, here, number one assumption. There is at least one cell population that is present in both batches. So basically, the Scanorama MNN was different from original MNN that batches with just single cell type, which is not present anywhere else, were allowed suddenly. And they actually do a good job. Actually, I value this method basically because of this experiment, because they actually generated this data set specifically for the paper. They generated this data set, cell lines, with like three batches, and the first batch contained only cell type 1, the second batch contained only cell type 2, and then the third batch had both cell types, and they show that Scanorama can, efficiently separate these two cell types based by, the clustering is with respect to cell type and not batch. And Surratt CCA and MNN, original MNN, they did a much worse job in their benchmarking. So I think that's a pretty nice demonstration, because in single cell, it's an unsupervised analysis. You don't know ground truth if you don't simulate your data or don't have this kind of controlled experiments. So it's very hard to know what is a successful integration. So how if you do dimensionality reduction or clustering or integration, it's an unsupervised analysis. You don't know whether it worked really well or not, because you have nothing to compare with. You don't know the ground truth. But here, at least, when you suggest a new method, you should, in my opinion, show either simulations or at least this kind of controlled experiment to demonstrate that really your method is really better than the other ones. And this is just my naive comparison of all these different methods using the data set that was provided in the exercises for this course. And here I'm plotting basically an uncorrected data set. So different colors, they correspond to different batches. And the uncorrected t-SNP plot, so you can immediately probably see the problem here. So for example, this pink cluster is driven by a single batch, and this is probably not good. So if you, it's unlikely that you can get one cell type to be present in only one batch. So probably this cluster is due to batch, not due to biology. And it's not well corrected by combat, as I said, since you can't preserve the valuable biological phenotype of interest in combat, in the combat procedure. So you can, yeah, it doesn't do a good job here. You still get this pin cluster driven by a single batch. And other methods, they actually do a decent job. So the single cell specific methods such as MNN, CCA, Scanorama, and especially Harmony. So Harmony is great at this kind of mixing all different batches across, mixing cells across batches so that all the batches are represented in all clusters. So visually it's maybe most appealing, but as I said, I have problems kind of intuitively grasping this, just algorithmic mixing of the cells. I am not sure that this is completely correct, but people like Harmony. I think Harmony has a good reputation. And then there were benchmarking papers claiming totally different results. So there was one wave of benchmarking papers comparing, here I'm kind of doing my empirical comparison. I don't know the ground truth here. So I assume that this pin cluster should not be driven by a single batch. But since I don't know ground truth, I can't be sure. And in these benchmarking papers, so people did a better job. They did, they were comparing these methods either in controlled experiments or in simulations. And so the first series of papers from Fabian Theis, for example, a big name in single cell, was claiming that you don't need to go beyond combat, that, just simple regressing out of your batch is fine. It's good enough. It's not outperformed by anything else, including MNN or CCA. So combat is just fine. And then the same author, the same Fabian Theis, was claiming something different a few years later, when it became obvious that the complexity of single cell data sets became so dramatic that indeed, when people realized that working with millions of cells, making this atlas level, atlas scale analysis, working with millions of cells, and working with, tens and hundreds of cell populations, and correcting for multiple batches, not just 1, 2, 3 batches, but maybe 100 batches. So it turned out that probably yes, you should go beyond combat. And basically in these benchmark papers, Harmony and Scanorama were demonstrated to work better than combat. And the reason is, again, it's about data complexity. So when Fabian Theis was claiming that combat is just fine for single cell data integration, at that time people were working with, just a few batches and a few cell populations and subtle batches. Then yes, if it's what is called low data complexity, then combat should be fine. However, if you have, a couple of hundreds, batches and cell populations, then probably you should apply something like this. Yes, so this was about horizontal data integration. So as I said, single cell people usually by integration mean this horizontal integration. So now I'm going to briefly cover vertical integration, which is integration across omics. So we are working with the same biological cells, but the feature names, they're different across data sets. So the idea here is that what was pointed out in one of the Surratt papers is that, single cell clusters based on single cell RNA-seq gene expression does not guarantee that these clusters are actually different, functionally different cell types. Because RNA-seq is just one layer of information, but maybe, if you add epigenetics, genetic, epigenetic information, maybe it will change this cluster that we observe. So this cluster is based on RNA-seq alone, but what if we add, proteomics or other types of information? Will the clusters change? Maybe, potentially combining different biological layers of information, we can discover more nuanced clusters. And so one of the first methods, single cell specific methods in this field was again suggested by Surratt, and it was a kind of CCA extension of CCA. They called it transfer anchors across omics. But essentially, if you look what they do, they start with CCA, canonical correlation analysis. So they take two data sets, one from RNA-seq, the other one from ATAC-seq, single cell ATAC-seq, and they just project these two data sets in the common axis of variation. This is what canonical correlation analysis does. And then they proceed with MNN. So if you remember the original CCA plus dynamic time warping, they did a CCA first, and then they did some additional correction with dynamic time warping. Here they kind of start with CCA and then proceed with MNN instead of dynamic time warping. But otherwise, they're kind of essentially working within single omic here. Because so as I said, this was early days, 2019. At that time, there were not so many technologies delivering multiple layers of biological data from the physically same cells. And that's why for this method, they actually use different biological cells and different omics. So ideally, this method belongs to what is called diagonal data integration, when the cells are different and the features are different. So that was super challenging integration. It was discussed in this Ricardo Argalago paper. Is basically there is no good strategy developed here at that moment. But Surratt still said, okay, yes, so the cells are different and the features are different, but we can at least map, single cell ATAC-seq peaks to gene names. So they kind of replaced ATAC-seq peaks with gene names, essentially bringing these two data sets back to the situation when you are working within single omic, within RNA-seq, and just integrating across, like bringing it back to horizontal integration, integrating across cells rather than across omics. But otherwise, it was quite successful for that time. So it was like one of the first attempts that I saw to integrate RNA-seq with ATAC-seq, and it was good, correspondence, well, visually, appealing ATAC-seq cells and RNA-seq cells. Remember, those were different cells, different biological cells, but they were kind of overlapping nicely. So it was clearly a very interesting method.
I had a couple of questions about the integration of Biomics. So if you go back one more slide. Yes, here. So, so from what I understand, they used different cells for our single cell RNA sequencing and single cell ATAC sequencing. But what they did is essentially they gave, was it a binary value or scale to the genes that were like showed chromatin openness with the ATAC-seq, or how does that work?
Yeah, so the interpretation, so I know how they technically did it. So basically they said, yes, the cells are different and the features are different, but we still, we can map ATAC-seq peaks to their, closest genes. Like you can group ATAC-seq peaks around the gene and say, from now on, the quantification is ATAC-seq, the height of the peaks, or how is it quantified? So the quantification comes from the ATAC-seq, but I call these features, a group of ATAC-seq peaks, grouped around the gene. I call it with a gene name. I assign to this, group of ATAC-seq peaks, I assign a gene name. And this is how I map, gene expression. So I have gene expression information from the same.
I see.
And now I have kind of another RNA-seq with just quantification from attack-seq. So
yes. So essentially, what they did is they identified where the attack-seq peaks were found, and if they're found, usually I'm guessing over like a transcription start site of a gene, then they assign it to that specific gene.
To the
And
best of my
then,
knowledge, yes.
yeah, exactly. And then basically what they did is they didn't know how to really integrate the attack-seq data, or I mean, they formed some clusters, but they didn't know whether it's true. So what they did is they overlaid it with the single cell data and saw that there is very good correlation.
Yeah, so here on the
Am I getting it
plot,
right?
so here on the sort of the consensus plot on the bottom, the red and the green cells, they are different biological cells. But in addition, the red cells, they are not, so the integration technically worked as if it was two RNA-seq data sets. But the quantification, like the values behind these red cells, they are actually coming from attack-seq. But they pretend, you pretend as if it was RNA-seq because you can make this mapping. You can group attack-seq peaks and assign them to genes. And you say, okay, let's forget that I previously worked with attack-seq peaks. Let's call them, let's call these features by gene names, by the names of the closest genes. And this is how you kind of, you can bring the situation back to already known case, to already previously established CCA plus dynamic time warping method. But they kind of still made one more modification. They removed the dynamic time warping and replaced it with this mutual nearest neighbor.
I see, but I think there's only one small thing. When you say different biological cells, do you mean like they're the same cell types, but in two, like, let's say two Petri dishes on which two different experiments were performed?
Exactly.
But they're still the same cell types, right?
It's the same cell type. Exactly. Yes, correct.
Okay, I got it.
Yeah, thank you for pointing this out. It was not, of course, cells from completely different tissues. It was
okay.
It was the same tissue, kind of. Just physically, it was different cells. While vertical integration, strictly speaking, this was one of the first slides. So vertical integration assumes that absolutely the same cell was used for extracting, genetic, epigenetic, gene expression information you extract it from exactly the same cell. But here the cells were different, but same cell type. Thank you for pointing this out.
Okay, thank you. That makes much more sense now.
All right. So here I'm listing just a few technologies delivering multiple omics from the same biological cells, such as site-seq, for example, sort of proteomics combined with gene expression. There was a nice technology, CNMT, that never became high throughput. So it delivered three omics from the same biological cells, transcriptomics, DNA methylation, and chromatin accessibility. But this was always, it's still, as far as I know, it still delivers only a couple of hundred cells, which is not, challenge for computational analysis, basically. And then multi-ohm 10x multi-ohm, providing gene expression and attack-seq information from the same cells is probably one of the best examples of really high throughput multi-omics technology. Yes, and then there are kind of quite a few methods developed in this area. If you look in the bulk, if you look in the bulk integration, then you can notice a few methods. So integration did not start with single cell. People tried to integrate different omics before single cell. And in this area, in the integrative omics area, there were quite a few directions already established that gradually came to single cell as well. So, for example, one of the most popular, and I would say like a standard, most of the successful methods, they do something similar. It's what I call convert to common space. What neural networks do, or autoencoders, And what UMAP and graphs do, graph-based integration, this is basically when you replace your original data, your original gene expression or ATAC-seq or proteomics data, you replace them with some abstraction, some graphs, and you are trying to overlap these graphs somehow. So I'll explain just in 1 minute. I'll explain it in more details. But this is one direction, very popular. The other directions, they were kind of less successful. So, for example, this canonical correlation analysis that I mentioned, even in single cell, you needed to do something in addition to canonical correlation analysis, such as dynamic time warping or mutual nearest neighbor on the top. But otherwise, it was sort of PLS and CCA was very old, very people with statistical background. They like this method CCA and PLS for integrating heterogeneous types of data. And then there was a very promising direction through Bayesian rule. So if you are familiar with Bayesian stats, basically, and think about Bayes' theorem, in the numerator of the Bayes theorem, there is a joint probability of observing multiple types of data. So the product of probabilities, so it's a priors and the likelihood, but essentially it's joint probabilities of observing multiple types of information. So it's an ideal setup for integrating heterogeneous types of data. But it has own challenges, some computational burden, etc., etc. So one of the hybrids between those two, between canonical correlation analysis and Bayes' rule, became especially successful and popular in single cell, and it was MOFA, which is also popular in bulk in bulk analysis. And I'll briefly explain MOFA. MOFA belongs to kind of you can think about MOFA as a kind of very how to say, it can really be related to generative AI even, because essentially it implements this factor analysis, which is generative learning, which is kind of so kind of generative AI is the next step. And there were two MOFA papers published. One was for bulk data. The first MOFA was published for bulk data, and then MOFA Plus or MOFA 2 was specifically targeting single cell data. I don't think they changed anything in the equations. So the math, to the best of my knowledge, in MOFA 2 is exactly the same, but they kind of made it more scalable, GPU compatible, and basically adjusted for operating with millions of cells. So I said that MOFA and factor analysis, they have something to do with generative AI. And the idea is very simple here. So you have observed data. You have observed data here on the bottom. And you are trying to reconstruct some hidden factors that generate this observed data. So, for example, you have three matrices: gene expression, methylation, and germline genetic mutations, and this is what the data you have. But you assume that all three of them, they came from the same biological process. And if you learn these two, latent factors that can generate the observed data, you can, you understand something fundamental about your sort of biological process, such as cell type or maybe cell cycle. These two factors can be related to, some very deep parameters in your biological process. In mathematically speaking, you can think about these two latent variables as mean and the variance. If you think about Gaussian distribution, there are two parameters, mean and variance. And if you learn these two parameters, mean and standard deviation, you can generate tons of samples from the Gaussian distribution. This is kind of an idea behind this generative model. That you learn some fundamental parameters that can result in, you can generate as much data as you want if you understand some fundamental parameterization of your process. This is what Morpho does technically. It's a figure from their paper. You can see here on the left-hand side, essentially, Morpho tries to decompose. So first of all, technically, factor analysis is, again, it has something to do with matrix multiplication and matrix factorization. That's why you kind of you represent all your original data sets through a product of two other matrices. These W matrices, they're individual for each individual data set, for each individual omic, and those are loadings. But then there is a common factor. So this is why, PLS and Morpho and CCA, the idea behind all these methods is actually construct this kind of common matrix that capture some common variation across omics. This is why it's called factor analysis. And here are your latent these factors. What is dimension? What's called factors is actually these latent variables L1 and L2. And then you can, Morpho provides a very, very interesting visualization. So it gives you this nice heat map where you can check what omics and how much contribute to each factor. So you kind of, first of all, you project your data on this common axis of variation, then you can check whether all three, in this case, omics contribute equally to each latent variable, or there is maybe two of them contribute more to factor 1, and then two other contribute most to the factor 2. And this is kind of, it's an interesting interpretation, interesting way to learn your data. So it's again a picture from their paper. Like technically, again, the idea was to generate this nice consensus plot by integrating RNA-seq and ATAC-seq. So now the cells are the same. So they say those are the same biological cells, just you visualize two layers, RNA-seq and ATAC-seq here, and this is your consensus plot. I'll skip this. Again, it's just an application of Morpho to this SCNMT, but it's again, it's a reiteration around the same things. So one important thing about Morpho I wanted to emphasize is that there is a very nice interpretation in Morpho. You can actually color how much each omic contributes to each cluster. You can create this interesting color map and show that maybe one cluster is driven by two omics only, but not all three of them, and the third one is contributing to another cluster. So it's actually an interesting interpretation. I'm not saying that it's maybe I'm not sure that it's very accurate, but it's an interesting way to think about the clusters. Maybe not all omics are contributing equally, but maybe there are some major contributions and minor contributions. So Morpho provides this kind of interpretation. So maybe the last important method I wanted to explain, I have still 2 minutes. It's a graph-based integration, graph intersection method, where the idea is, again, simple. You construct KNN graphs for 3 individual omics. So here the nodes are cells, and those are the same cells. So across all these 3 graphs, you have exactly the same cell IDs, but the edges, the connections, they were constructed using single cell RNA-seq, single cell bisulfite-seq, the methylation data, and single cell ATAC-seq. Once you, so first of all, a nice thing about graphs is that you can specify, appropriate distance. So for example, for RNA-seq, maybe Euclidean distance is good enough, while for ATAC-seq and bisulfite-seq, those are binary data, essentially 0, 1. Maybe Hamming distance is better suited for binary data and not Euclidean distance. But once you computed these graphs, these graphs, they don't remember anything about the original technologies they were produced by. So now you can use this trick, which is graph intersection, which means you keep consistent edges. If the edges were consistently present across 3 different omics, 3 different graphs, they propagate to the consensus plot. So basically, these 2 guys, they were connected here in the omic 1 and in the omic 2 and in the omic 3. So you propagate this edge down to the consensus plot. This is what is called a graph intersection, and this is what can be implemented actually in UMAP with just one single line of code. Because remember that UMAP starts with graph construction, so it constructs high-dimensional graphs, and then it's absolutely natural to actually, and there is a means in UMAP with just one line of code, you can, first of all, you build 2 high-dimensional graphs for individual omics, and then with just one single line of code, you can overlap them, and you get this beautiful consensus graph, which is kind of a combination of RNA-seq and single-cell proteomics. So it's a side-seq benchmark data set from Surat. And this is what was actually implemented later in Surat weighted nearest neighbors. So basically, if you check the manuscript and the equations, it's basically, it's literally graph intersection. So they construct KNN graphs and then compute a consensus. They construct KNN graphs on individual omics and then compute a consensus graph. And again, the same can be done within autoencoder. So I already explained how the autoencoder works. And neural networks and autoencoders, they allow this natural data integration because you can provide individual omics to different layers and merge them somewhere in between. And neural network is essentially a graph. So it's kind of the same idea again, sort of a graph intersection idea, and this is the result. So the result for side-seq looks very similar to what you got from UMAP, and so on and so on. This AI ideas, they became super popular in single cell. And for example, this SCVI, what I highly recommend, what I used. It's a good tool for data integration, and SCGPT. Published a few years ago, also kind of promising generative AI thing. To the best of my understanding, so when I read this paper, it's again about kind of fancy autoencoder. So it's a data integration essentially tool to me, although they kind of propose to see it in a different angle. And so on and so on. It has something to do with data, with the amount of data in single cell. So we are really talking about, millions of cells nowadays. Big labs, big, consortia, they are working with millions of cells, and it's a big data. And if you have big data, you can do AI. You can go beyond linear analysis. You can do all fancy non-linear machine learning algorithms. And in my understanding, so I'll skip this. So basically, the advantage of single cell data is the sample size, which is clearly outstanding compared to other areas in life sciences. Thank you very much for your attention.
Yes, thank you very much. It was very intense but very interesting statistics and comparisons. I have more like practical questions, like in 10x multiomics, you don't need to do integration because they come already with the same cell barcode, right? So I guess that you don't have to integrate. That's the first question, but then it's going to follow up with the fact that then if you have 2 different samples that have run through multiomics from 10x, and I don't know, same cell type, though, but different time or different labs even. So what do you integrate? Only the transcriptomics, or how would the integration would work there?
So for 10x multiome, you get ATAC-seq and RNA-seq from the same cells. So
yes.
It's yes, you should have some commonality. When you do integration, something should be the same across data sets. Either the features or the same cells. So here you have cells that are the same. So the integration is actually to use cells as anchors to combine RNA-seq and ATAC-seq and generate this consensus, you can say consensus t-SNE or UMAP plot that is a result. It is exactly the same cells, but it's a result of not just RNA-seq, but RNA-seq plus ATAC-seq. That's the integration. So the goal is that you should have one thing in common always. When you do integration, unless it's diagonal integration. For diagonal integration, you have both features and cells different. But while this vertical integration with 10x multi-ohm, you should have cells with the same barcodes. Like, well, it should be like the same cells. What you are integrating different biological variables. You're integrating ATAC-seq and RNA-seq.
Yeah, in the 10x you have the same barcode for ATAC, for the libraries of ATAC and the libraries of transcriptomics. It is the same barcode.
It's the same cell. It's the same cell. So cell has two biological variables. So each cell has quantification of ATAC-seq peaks and gene expression.
Yes.
And this is what you're integrating.
Exactly. Then it's already it is already integrated somehow. But then you bring
integration doesn't mean integration. So these two biological variables are not integrated.
No, but I mean when you do the analysis, I mean there is like an anchor that is the cell barcode. The same cell has two informations, right? Two data sets, the same cell.
It will give you like each of them will give you a separate UMAP. What you want to have, you have a consensus UMAP.
Okay.
Which includes, RNA-seq plus ATAC-seq, not just RNA-seq alone.
Yes. And then there's a follow-up question. So if you bring another data set with the same multiomics, but in a different lab, do you have to integrate the 4 data sets, or is it enough just to integrate transcriptomics and then you use overlay the ATAC-seq?
Yeah, so here you are doing kind of both vertical and horizontal integration, and should be probably done step by step. So you start with first horizontal integration, integrate, RNA-seq across different batches, and ATAC-seq across different batches, and then you proceed with, integration of actually features, ATAC-seq and RNA-seq. But the good thing is that you have anchors. So when you have anchors, so if cells are the same or features are the same across different data sets, then this is straightforward. This is what allows you to do integration.
Okay.
Otherwise, the worst case is actually if you get different omics and different cells, even from the same tissue, but biologically different cells, or let's say physically different cells produced by different labs. So the features are different, the cells are different. This is a diagonal integration. You don't have anchors anymore, and then this is a problematic case. You don't know how to get this the single consensus UMAP.
Okay, thank you, Nikolay.
You're welcome.
I have a question regarding the data integration of different, based on like technical aspects. Suppose like I have like a data set which contains like 10,000, 20,000 cells, which has like 3 or 4 clusters that I have found out from my own analysis. And then I have a bigger data set, which has like maybe 400,000 or something. Does that, with like maybe 5 or more clusters, does that affect like during the data integration process? In that case, should I subsample the more bigger data set and then run the integration analysis, or?
Yeah, so here's so what I just explained, all this integration that just explained. So it's supposed to kind of merge data set. When you have one data set dominating, if you have a reference atlas, maybe you don't need to merge. You don't need to download, this reference atlas and merge it with your tiny particular data set in order to see where your cells are clustering. But the more common approach is actually to project, to build any machine learning classifier. You take this atlas, you create, multinomial regression, like extension of logistic regression. You create any machine learning classifier that can assign your query cells from your particular data set to this atlas. That would be probably even more, I would say, even more accurate approach. Because indeed my intuition is that when you integrate data sets that are dramatically different in size, then there can be some, issues. I think all these integrative methods, they are designed for comparable data sets. Well, there is another integration approach where people project, and I think you single cell people, you all know about this. Just I haven't touched it here because my explanation was around sort of merging data sets. When you really want to merge two data sets for any purpose, then you use this method that I just explained. But if you if you want to annotate your cells from your query, small data set, if you want to just quickly check what kind of cell populations are present in your query data set, and you have big atlas, so it doesn't, you don't have to merge those two and create the consensus UMAP. So there is a simpler way of projecting.
Okay, thank you.
You're welcome.